Hello, welcome to a lecture on the kidney and body fluids. I am Dr. Eric Edu. I'm with the Department of Anatomy and Cell Biology of the School of Medical Sciences, University of Cape Coast. So that is a micrograph which has been enlarged. So when you look at this, it's actually taken from this vertical section. So you are only looking at a lobe of the kidney, okay, with the renal papilla showing here. So that's the whole lobe. Then you can see that within this lobe, you have a cortex and then a medulla. And they have a cortex and a medulla. The medulla is divided into an outer medulla and then an inner medulla. So that's the renal lobe conformation. Then the kidney cortex and medulla. So again, these dashed lines are demarcating the medullary region and then the cortical region. The cortical region will contain the pars convoluta and then the renal corpuscles, which are almost the same. Renal corpuscle, pars convolutus, we see because it is convoluted, usually because of the appearance of the glomerulus. It looks convoluted. Okay? But the glomerulus together with the Bowman's capsule will become the renal corpuscle. Then these are the straight running collecting tubules that form the medullary rays in the cortex. The AV stands for an arcuate vessel, which is very important in demarcating the region of the cortex from the medulla, usually. So the pars convoluta or cortical labyrinth contains the renal corpuscles and the convoluted portion of the glomerulus. And then also you have the medullary rays, also called the pars. So the red arrows are in the medullary rays, as you. So the tubules also have convolutions in addition to that of the glomeruli, as in you have the proximal convoluted tubule and then distal convoluted tubules. So those also will appear as a cortical labyrinth. All right. So you can see that this is a bigger size of the kidney, and then you are looking at a maximized portion of it. This is more or less one of the lobes, as you see. So once again, you have the renal corpuscles and then the convoluted tubules, pars convoluta. MR will stand for the medullary rays, then the interlobular artery and renal corpuscles. It's important to note this, that the medullary rays, also known as the pars radiata, is made up of the collecting duct, the proximal convoluted tubule, and the distal convoluted tubule. These three have been labeled A, B, and C. It's our duty now to be able to assign them names. What would this stand for? What would that stand for? And what would this also stand for, the A, B, and C? So I'll give you a clue. You can see that with the A, you have the lumen being eosinophilic, okay, and it's because of the presence of some microvilli within it, okay. So that is one of the striking difference between A, B, and C, because C and B are both hollow without any stain within it. That now it is left with the C and B. How are you going to differentiate between the two? You can also tell that B's diameter is smaller than that of C, so C is wider than that of the B. Okay, so the C is the collecting duct, and B will be distal convoluted tubule, and then A will be proximal convoluted tubule. So that is the emphasis. So that's the way you differentiate between them. Okay. Also, the cuboidal nature of the lining epithelium is very clear in the collecting tubules compared to the others. This looks more squamous in nature. That's as that one too. But this is very clear cuboidal. So when we look at it in the transverse section too, this is what we see, a larger lumen for C, which is the collecting tubule, with this clear cuboidal nature of lining epithelium. Then you have that of the distal convoluted tubule. And remember, both the collecting tubule and distal convoluted tubule will not have an eosinophilic or stained lumen. All right. So that is one of the proximal convoluted tubule. And you can see that there is haziness in the lumen. The lumen is not devoid. It is actually stained because of the brush border we talked about earlier on. So you can as well count a lot of the proximal convoluted tubules. So there's another one here. You have some there. You have some there. Then you also have some of the distal convoluted tubules there. Then you have the collecting tubule. So you can work them out. This is also a collecting tubule. So those are the emphases. So this describes the blood supply of the kidney. So we can start our journey from the renal artery, which goes inside and forms segmental arteries. Then the segmental arteries also go ahead to form the interlobar artery. And then from the interlobar artery, you get your arcuate arteries, which usually separate the cortex from the medulla. It's one of the landmarks you use to tell. Then arcuate arteries will further branch to form interlobular artery. Then the interlobular artery will branch to form the afferent arteriole, which is also known as the intralobular artery. Then you form the glomerulus and then the efferent arteriole. So that will now join, you form the peritubular capillaries associated with the convoluted tubules. And then you form the vasa recta, which is associated with the nephron. Vasa recta because they run straight. Then you have the interlobular vein, the arcuate vein, interlobar vein, and then you have the renal vein. So that is what you see there. So those will be the peritubular capillaries, and that will be the vasa recta. So the vasa recta is usually found in the medulla. The peritubular capillaries are usually found in the cortex. If you take a lobe, which will be found in the lobe, this is what you see. So that will be the pars radiata with the medullary rays, intralobular artery, which will be the afferent arteriole. Then you have interlobular artery in between the lobules. So again, you can see all these pars convoluta containing glomerulus, and then you may also find convoluted tubules elsewhere. Then that will be the pars radiata. So medullary ray arcuate artery is AA. Interlobular artery. So inter and intralobular artery. As I said, the intralobular is also known as the afferent arteriole. That is the urinary pole. You can easily tell that that's a Bowman's capsule. So urinary pole, vascular pole is expected to be opposite. So that is the vascular pole. That you have urinary pole here. So that is a glomerulus. All these are some of the collecting tubules. So the parietal layer of the glomerular capsule consists of a simple squamous epithelium. So that's the parietal layer, simple squamous epithelium. At the tubular pole, this epithelium changes to the simple cuboidal epithelium, characteristics of the proximal convoluted tubule. So once you get it, once the parietal continues as a proximal tubule, it changes from squamous to cuboidal. Already we have established that the visceral layer will differentiate to form a special layer known as the podocyte, which is in close association with the glomerulus. So that is afferent arteriole, and that is efferent arteriole. And then that is distal tubule with macular denser cells, that's the lumen. Then at this portion, you have lacy cells. You see them in a clearer picture later on. Then within the afferent artery, you have these specialized smooth muscle cells known as the Juxtaglomerular cells. Then within the glomerulus, you also have Mesangial cells. Mesangial cells. So the podocyte that is found between the visceral layer of the Bowman's capsule and then the glomerulus, this is what happens. So you start having primary food processes here. Okay, so that's a podocyte. And then it develops primary food processes, which will also branch to form the secondary food processes, and within which you get the filtration slit. So that is the glomerulus, and it is fenestrated. So when blood passes through, and because of the high pressure within the glomerulus, certain fluids and cells are forced out of the glomerulus, and those that are unable to pass through the fenestrations will go on with the circulation and come out in the efferent arteriole. So the substances that are able to pass through, especially the fluid portions of the blood, will go through the slit. So it's just like sieving something at home in the kitchen when we do palm soup and you have to pound the fruit. You end up having chaff together with what you really need. So you mix it with water and then pour everything in a sieve. So what you need is just like the ultrafiltration, is what will go into your saucepan that you use for your palm soup. So that siever is just like that of the podocyte network that we see here, together with the glomerular fenestrated capillaries. So during embryonic development, the simple epithelium of the parietal layer remains relatively unchanged, whereas the internal visceral layer is greatly modified. The cells of this layer, the podocytes, have a cell body from which arise several primary and then secondary food processes, also called pedicels. Okay, so that's the endothelium. That's the basement membrane of the endothelium. And then you also have the basement membrane of the podocytes. Okay, together they are back to back. Basement membrane of the podocyte and that of the endothelium are attached together. Okay, and then you have the endothelial lining itself of the podocyte. So this is also basal lamina, that is endothelium. Remember, when you say lamina, it has to go with basal. When you say membrane, it has to go with basement. That's primary food process, secondary food process, that's the podocyte. That is the modified visceral layer of the Bowman's capsule. So identify the different cell types in this diagram of the glomerulus. The arrows indicate the nucleus or nuclei of the cells in question. So you should be able to tell those. The podocytes and then endothelial cells. So that is the glomerular capillary, and then that is the podocyte. So that will be a filtration barrier. Okay. You have the endothelial lining, the basement membrane of both the endothelial lining and then that of the podocyte, and then also the podocyte's epithelium. That's the glomerular mesangial cells. In another scanning electron micrograph, you find podocytes with the cell body and have primary food processes with the arrows showing, and then secondary food processes. So again, you have the endothelial cell. We are looking at the glomerulus. So that is the lumen, that is the basal lamina, and obviously that will be the endothelial lining of the glomerulus with the fenestrations. Those are spaces which allow easy flow of fluid out of it. Then to pass through the pedicels, that's the secondary food processes, and then it will go into the Bowman space or the urinary space, as we said. And then it will go through the proximal tubule. Before then, you know, the urinary pole is also there. So it will receive it and then go to the proximal tubule, and then it will continue its journey down the tubule through the descending limb and ascending limb of the loop of Henle to the distal convoluted tubule, then to the collecting tubules and collecting ducts. So this is an expanded form. That's the basement membrane of the two of them. That's the podocyte and then the endothelium. So this region is that of the endothelium of the capillary. So that will be the endothelial lining. Those are fenestrations. That's why you see a pore there, another pore here. And these are the primary food processes or pedicels. So that one also has spaces in between them, filtration slits. Okay. So these are some important structures we need to bear in mind. Juxtaglomerular apparatus, GGA, is a specialized, smooth muscle cells in the afferent arteriole. So that is the juxtaglomerular cells, smooth muscle cells related to the afferent arteriole. Then you also have the juxtaglomerular apparatus, which is a specialization of the afferent arteriole and that of the distal convoluted tubule of the same nephron. It is involved in the regulation of blood pressure through the renin angiotensin aldosterone mechanism. So the juxtaglomerular apparatus is made up of three components. You have the macular denser cells, juxtaglomerular cells, and then extraglomerular mesangial cells. So these are the structures we are talking about. So that is the macular denser cells here. Then you find the juxtaglomerular cells. These will be lacy cells. Those lacy cells are found between the distal convoluted tubule and, for that matter, macular denser cells and that of the afferent and then efferent arteriole. You also have other specialized cells. So extraglomerular mesangial cells are also known as lacy cells. So you have this distal convoluted tubule, that is the macular denser cell, lacy cells, then juxtaglomerular cells, that will be the afferent arteriole. Obviously, this will be the podocyte and then the glomeruli, and that will be the urinary space or the Bowman space. That will be the urinary pole, and then that will be the vascular pole. Then this is the parietal layer of the Bowman's capsule with the squamous cells. Then that will be the proximal tubule that has the cuboidal cells. Then obviously those tubules we looked at distal tubules and then also the proximal tubules. And we know that this will be more likely to be proximal, and that is more likely to be, yeah, most of them are even hazy. A lot of them likely to be actually proximal convoluted tubule. Mesangial cells, what are their functions? They are for physical support and then contraction. They also have phagocytose substances like protein aggregate that adhere to glomerular filters, including antibody-antigen complexes. Then for secretion, the cells synthesize and secrete several cytokines, prostaglandins, and other factors important for immune defense and repair in the glomerulus. So this is very clear, proximal convoluted tubule. So like I said earlier on, this is likely to be another proximal convoluted because most of them have their lumen, stained. That is a clear distal convoluted tubule. So the reason why the proximal tubule is stained or looks hazy is because of this microvilli. Okay, so that is why you have that. That's a collecting duct. That is a distal convoluted tubule or distal tubule. That will also be another proximal tubule because of how it is stained. So usually the only confusing aspect will be how to differentiate between the distal convoluted tubule and collecting duct. Okay, but you can always use the diameters to tell. And then also the clear cuboidal nature of the cells. Okay. So you can see that these are very clear cuboidal and then wider lumen compared to this. And this is also more oval in shape or has a little collapsed lumen compared to the more or less circular nature of that of the collecting tubules and obviously proximal tubule. So in summary, we have looked at the kidney and then the fact that you have the nephron made up of the renal corpuscles and then the tubules. And that we said that the corpuscle is made up of the glomerulus and then the Bowman's capsule. We also mentioned that the Bowman's capsule has a visceral layer and a parietal layer, and the visceral layer is specialized to form the podocytes. And the parietal layer is squamous in shape and continuous as a proximal tubule with cuboidal cells. And with the podocyte, which is highly invested with the glomerulus, it has the filtration barrier being the endothelium of the capillary, the basement membrane of the capillary, and then the epithelium of the podocyte itself. We also said that the podocyte has a primary food process and then secondary food process, which is known as pedicels with filtration slits. We also looked at the proximal convoluted tubule, distal convoluted tubule, and then also the loop of Henle. We have mentioned that the Proximal tubules are usually stained, unlike the distal tubule and then collecting tubules. So basically, that is what we have covered today. Thank you very much. Hope this video helps.